Okay, in this video, we're going to talk about the biological carbon pump. This is a really important process that affects the marine carbon cycle, the amount of carbon in the atmosphere in a number of important ways. And so it's worth spending some time on. In this video, we'll trace the vertical distribution of carbon in the ocean. We'll explain how phytoplankton influence atmospheric CO2 levels. And then we'll discuss how changes to the biological pump can affect climate. So in the last few videos, we've talked a lot about this air-sea exchange of CO2. Every time a wave breaks, carbon in seawater is going back to the atmosphere as CO2. At the same time, there's air trapped in little bubbles in the sea foam here that's exchanging and returning and dissolving into the ocean. So this cycle of dissolution, escape, dissolution, escape reaches this chemical equilibrium in the upper ocean. You can think about this upper ocean as being around 100 meters deep, which is essentially the winter mix layer. So how deep that part of the ocean will mix over an annual cycle. And then it's up to ocean circulation to exchange that mixed layer water with the deep ocean below it that's not actually in contact with the atmosphere. And so while this air-sea exchange of carbon is occurring, there are phytoplankton growing in the ocean that are taking inorganic carbon and converting it into organic carbon to form their biomass. Now, of the total amount of photosynthesis that occurs in the euphotic zone, most of that is either respired back to CO2 by phytoplankton themselves as they build their cell walls, their DNA, their proteins, and much of the rest is consumed by the things that eat the phytoplankton. These are the zooplankton. These are protozoan grazers. These are bacteria that live in the ocean. They're taking the biomass, consuming oxygen, returning that carbon back to CO2. And that's the story for around 85% of the photosynthesis that happens in the ocean. But around 15% actually sinks below this euphotic zone before it's respired. And this might not necessarily be living phytoplankton anymore. This could be zooplankton fecal pellets. This could be just dead guts of diatoms that aren't actually grazed by zooplankton. These tend to stick together and form these big aggregates called marine snow. Because it's all glommed together, a bigger particle, it'll sink faster. And so as this marine snow starts sinking to the seafloor, only around 0.4% actually makes it to the seafloor. The rest is eaten by animals and bacteria in transit. And as this carbon hits the seafloor, a whole new set of organisms starts to act on it, and most of that is consumed. And in the end, only around 0.1% of the original photosynthesis ends up staying in sediments. We refer to this carbon as buried. It's essentially removed from the ocean, air, and land carbon cycle, this rapid carbon cycle. And the only way it will come back is through the slow carbon cycle. And so this whole process, this production of biomass in the surface ocean, the sinking below the euphotic zone into the mesopelagic, into the abyss, and its eventual consumption by bacteria and zooplankton is what's called the biological carbon pump. And really what it's describing is a removal of carbon from the surface ocean and its transport into the ocean interior. We can see this effect by looking at the profile of dissolved carbon against depth. And so if you sample the deep ocean for dissolved carbon, you'll find there's a lot more than in the surface ocean. In the surface ocean, there's only around 84 milligrams of carbon per liter, where it's over 100 in the deep ocean. This 84 milligrams is actually what's in equilibrium with atmospheric CO2. So atmospheric CO2 is really reflecting the surface carbon concentration, not this greater value in the deep sea. So we can see additional signatures of this. As zooplankton and bacteria are consuming this biomass, it should also consume oxygen. And so concentrations of dissolved oxygen also decrease below the euphotic zone as this biomass is respired by the organisms that depend on it. And so you can imagine a scenario where there's no biological pump, where ultimately the effect of mixing will homogenize the carbon concentration. So it's the same concentration in the deep ocean as it is in the surface. And as this equilibrates with the atmosphere, the value in the atmosphere you'll find is over 750 ppm, over 3 times the pre-industrial CO2 value. And so really it's only because phytoplankton growth and that sinking of that biomass to the ocean depth is constantly removing carbon. That's the reason why there's not that much CO2 in the atmosphere. So for instance, if you take seawater from around 2,000 meters down in the water column and you allow that to equilibrate with an atmosphere, the amount of CO2 that you'll find is around 750 ppm, which is much higher than the 207 ppm of the pre-industrial atmosphere. So the sinking of phytoplankton biomass to the deep sea allows more CO2 to dissolve into the surface ocean, which is then taken up by phytoplankton again, which sinks to the deep ocean and then is returned to CO2. And this process overall gives us the modern carbon dioxide concentration of 270 ppm. Ultimately, the atmosphere is in equilibrium with around 84 milligrams of carbon per liter, but why not less? Well, the answer is that the nutrients that power phytoplankton growth have run out at this point. You can see the profiles of two nutrients, phosphate and nitrate. In most regions, phytoplankton nutrients like nitrate and phosphate are depleted in the surface ocean. So they're totally exhausted by the phytoplankton because at some point in the past, phytoplankton have grown here before, their biomass has sunk down to the ocean depth, and it's been returned to CO2, and these nutrients have been regenerated. But since there's no nutrients left in the surface, the amount of photosynthesis that can be supported is a lot lower. So that as a result, the biological pump essentially stops. And this is the condition for most of the ocean. You can see throughout the tropical Atlantic Ocean, the Indian Ocean, and most of the Pacific Ocean, there's no nitrate in the surface ocean. You see these really purple colors indicating that nitrate concentrations are close to zero. But there's three major regions that have unused nitrate. There's the Southern Ocean, there's the Equatorial Pacific, and then there's the Subarctic Pacific, the North Pacific. And so for these polar regions, we can try to rationalize this. We know that the growing seasons are short and that light, especially in the wintertime, is low. And so maybe phytoplankton can grow as efficiently as they would be able to in the tropics. But that doesn't always explain, for instance, in the Equatorial Pacific, there's lots of light. The growing season is essentially 12 months out of the year. So why aren't they using these nutrients here? So we'll come back to this question in a minute. At the same time, we can look at ice core records of atmospheric CO2, which suggests that the biological pump was more efficient in the past. So as glaciers grow, there's often air bubbles that are trapped inside the ice. And in that air, there's a record of what carbon dioxide was at that given point in time when that ice formed. So this is a record from an ice core over 800,000 years showing atmospheric CO2 and reconstructions of temperature based on isotope proxies. At the top of this graph is the pre-industrial atmosphere of the Holocene, for roughly the last 10,000 years. 20,000 years ago, CO2 was much lower, only around 180 parts per million. And it was at a similar level throughout much of the last 100,000 years before jumping up to around 280 again. Before that, it was lower. Before that was high. Before that was low. Before that was high. This cycle of low CO2 and high CO2 intervals corresponds to global temperature. This is a temperature record from Antarctica. You can see that low CO2 occurred when temperatures were also colder. The high CO2 corresponds to warmer intervals. These colder, low CO2 intervals are the ice ages. They occur frequently and regularly over the last million years of Earth's history. During this time, global temperatures were around 6 degrees cooler than they are today on average. And the terrestrial biosphere was quite different. You may have seen fossil skeletons of woolly mammoths, mastodons, and a whole bunch of other Pleistocene mammals that are no longer around today. The main suggestion for why these organisms aren't around today is due to this climate change right at the end of the last glacial maximum, at around 20,000 years ago, into the Holocene of today. This dramatic rise in CO2 and this rise in temperature. So today, ice sheets only occur in Greenland and in Antarctica. But during this last glacial period, they extended throughout much of the continental US, all of Canada virtually, the Alaskan Peninsula, points through Siberia, tall mountain regions like the Himalayas, through parts of Russia, much of Northern Europe. So these ice sheets grow from ocean water that's been evaporated in the atmosphere, snows down in these polar regions, and stays there throughout the years, eventually becoming ice sheets. And during this time, so much of the seawater is locked into these ice sheets. The sea levels actually fell 100 meters or so. This is what allows this land bridge between Siberia and Alaska to be accessible. So this is a really dramatic change in Earth's climate that's associated with really low CO2 values. So what keeps these CO2 levels low? In the last video, we estimated the residence time of the rapid carbon cycle is around 90,000 years or so. So that's too long for most of these geological sources, whether they're volcanoes adding CO2 or the burial of carbon in marine sediments, to explain this. These changes are happening too fast, and so this has to be due to a reorganization of carbon in the cycle between ocean and land and atmosphere. At the same time, you'll remember that forests and soils, they turn over the carbon really fast, so it's unlikely that these will be places to store carbon for thousands and thousands of years. So this is a head scratcher for a while, but further ice core measurements helped establish a new hypothesis. So this is a zoomed in record of the atmospheric CO2 change in ice cores over the last 120,000 years. Around 20,000 years ago was its last glacial maximum. We're here today in the Holocene at around 270 ppm CO2. During these cold periods, these glacial cycles, these ice cores have lots of dust in them. And so their iron contents, iron being around 3% the mass of continental crust or dust, the amount of iron in these cores increases a lot during glacial periods. But iron is also a nutrient to phytoplankton. And so from this comparison, one might suggest that the biological pump might become more efficient due to this extra delivery of iron. So at this time, there had already been some measurements of iron in seawater, but it was slowly being realized that those measurements were contaminated by the fact that these measurements were taking place on boats that were rusting really fast. So I don't know if you spent time out at sea, but you'll notice that everything turns to rust remarkably fast. If you live in a coastal region, your car will rust really fast, and this is due to the amount of salt in the air. And so for decades, this was contaminating people's measurements when they wanted to measure how much iron was actually in the seawater. After this was realized and overcome, it was understood that seawater itself is really depleted in iron. Iron isn't very soluble in ocean water. At the same time, studies of plants had indicated that iron is really important to photosynthesis. The actual photosynthetic apparatus requires 23 iron atoms, which is a lot. And so studies with plants grown in soils that had all the iron removed showed this phenomenon called chlorosis. You can see here is a leaf like this, where instead of being bright green, this yellow color remains due to the fact that the cells can't make chlorophyll because they can't photosynthesize. So maybe this is happening in the oceans. And so one more piece of evidence came from satellite maps of dust in the atmosphere. You can see areas of high dust occur overlapping with the largest deserts of the world. Sahara Desert is a major producer of dust that gets blown out to the Atlantic Ocean. You'll notice too that the Atlantic Ocean has all of its nutrients consumed. In contrast, places like Antarctica get almost no dust deposition during modern times. This correlates with the high nitrate that's observed there. So it would suggest that the iron supply is too low to consume all the other nutrients that are in the seawater. You can see similar overlaps here in the North Pacific. There's lower dust and leftover nitrate. And in the equatorial Pacific, you can see this is also a low dust environment as well. And so correlation doesn't mean causation, but it's enough to form a hypothesis. Does iron scarcity actually limit the efficiency of the biological pump, this ability to remove nutrients and carbon from the surface to the deep sea? So to test this hypothesis, this is where seawater is scooped up, put in a bottle, and then allowed to sit in the sunlight for a few days. In some of these bottles, the experimental controls, nothing else is added. But in this example, in the North Pacific, iron was added to different amounts. And the different amounts of iron are colored by the symbols here. You can see that the amount of chlorophyll recovered after 5 or 6 days or so is a lot lower in these squares. These are the 0 iron treatments, so no iron is added here. In contrast, every experiment where iron was added had more chlorophyll after a week of incubating in the sunlight. At the same time, nitrate and the other nutrients that were measured in the seawater were drawn down, were removed in the treatments where iron was added. So this suggests that iron limits the growth of phytoplankton in the ocean. But at the same time, these are just bottles. They're a couple liters at most. And so try as you might, you won't fit an entire ecosystem into a bottle like this. You'll capture a lot of microscopic phytoplankton, sure. You'll capture bacteria and viruses, but the zooplankton that are swimming up and down hundreds of meters every day, fish, everything else isn't going to show up in these bottles. It's just too small. And so the next step was to conduct similar experiments, but out in the ocean. These are called ocean iron fertilization experiments. So we'll walk through these plots, but they're basically showing distance in both directions. So this is distance north and south. This is distance east and west. And each of these plots corresponds to a day of the experiment. This top row shows an inert tracer. And so in these experiments, iron is being added to seawater by dumping it overboard in barrels. And it occurs with this inert tracer, just something they can measure to know where iron is added to. And the reason is because some of this iron will be taken up by phytoplankton for their growth. Some of it will just turn to rust and sink to the bottom of the ocean. And so you want something that won't react to tell you where the iron was added as the ocean is stirring itself. So over about a week or so, you can see this enriched patch of this tracer start to dissipate and mix outwards. And then at the end of the experiment, it's not detected at all. You can see iron in this second row Iron is added and then it decreases. It's actually added again, decreases, added a third time and decreases. And then it just stays low at the end of the experiment. So what about chlorophyll? Iron is added. Will phytoplankton respond? Will the chlorophyll go up? After 5 days, you can see there's a lot of chlorophyll in this patch. And after 8 days, there's even more. After 9 days, there's even more. And then all the iron is consumed. And so the bloom subsides. At the same time, this panel is showing the amount of nitrate that's been taken up by the phytoplankton bloom. And it's quite a lot. So it seems like the results from the bottle incubation also occur out in the ecosystem when the entire marine ecosystem can respond. And lastly, this row shows CO2 uptake. So in the iron fertilized seawater, you can see that CO2 has been removed from seawater. Phytoplankton have taken up CO2, they started to sink, and this has removed carbon. So this suggests that the biological pump in these high nitrogen regions is limited by iron. And going back to glacial times, we know the atmosphere was dustier. That's what those ice core records were showing. And so one of the best explanations for the low CO2 found during glacial periods is that the ocean's biological pump had become more efficient. And so surface carbon dioxide was much lower than it is today. And this helped maintain a low atmospheric CO2 inventory for thousands of years. So to summarize that, you can compare the oceanic carbon cycle of today versus the last glacial maximum. Today, we have a comparatively larger atmospheric CO2 reservoir. It's controlled by the carbon content of the upper ocean. Now, mixing between the deep ocean and the upper ocean occurs every 1,000 years or so. Over that time, this biological pump is removing CO2 from the surface into the deep ocean. This biological pump seems to have become stronger during the last glacial period, and so this makes the removal of carbon from the surface to the deep much more efficient. At the same time, there seems to be a lot less mixing between the deep ocean and the upper ocean. And a lot of this has been related to the extent of sea ice during this period that is basically capping the ocean, and it's limiting the impact of the winds to really stir up the water. So as a result, you have the ability to store a lot more CO2 in the deep ocean that isn't exchanging quite so fast with the upper ocean. And so the carbon difference between surface and deep becomes more intense in the glacial period than it is today in an interglacial period. And this reduces the atmospheric CO2. And so we might be tempted to use the same processes, this iron fertilization that occurred naturally during the last glacial period, as a solution to climate change. This is a way to remove CO2 out of the atmosphere and store it in the ocean, right? And for a while, this is a real possibility, but eventually some key questions emerged. And these questions really haven't been adequately answered since. For instance, if you keep dumping iron into the ocean, how are you changing the ecosystem? You're probably selecting for a different group of phytoplankton to grow than would otherwise be there naturally when iron was a limiting nutrient. How does that affect the organisms that feed on that phytoplankton, that feed the fish and the mammals that live in those waters? There's another big question of, does it actually work? What these experiments tend to show is that the phytoplankton respond in the surface layer, but it's actually quite difficult to quantify how much has been removed from the surface and exported to the deep ocean. And in some of these experiments, the marine consumers, the zooplankton and bacteria, respond just as fast as phytoplankton. So even though you have more photosynthesis happening, that's being eaten much faster. And so the amount of carbon that is available to sink doesn't change. And there's a related question of how long this carbon will be stored. Even if a significant fraction of this new production sinks below 100 meters, a lot of it will be eaten by 200 meters, even more by 300 meters. That carbon is more likely to be mixed back to the surface ocean due to storms, eddies, and other mixing events. And so in that case, it's not actually sequestered at all. And so because of these questions, there's been a moratorium on ocean iron fertilization studies for the last 10 years or so. It's illegal to dump iron in the ocean by international agreement. In the last few years, these questions have been highlighted due to this rogue geoengineering experiment where iron was illegally dumped off of British Columbia. The goal for this was to increase the amount of salmon available to the indigenous peoples of British Columbia, but there's no clear indication that it worked. This increase in satellite chlorophyll here is attributed to the iron fertilization effect. But you can also see that these coastal plumes of phytoplankton production are common just by mixing of coastal iron out to sea. And so it's hard to be sure that the iron fertilization here and not mixing of coastal waters is what caused this bloom here, these high chlorophyll patches. And this would be a big problem for any geoengineering scheme that has to prove that it's sequestering carbon. So to wrap up, In this video, we've summarized the ocean biological carbon pump. We've traced the vertical distribution of carbon in the ocean and explained how phytoplankton contribute to this shape by taking CO2 out of the surface ocean and shuttling it to the deep ocean. In this process, they're influencing atmospheric CO2 because that's what's in equilibrium with the surface ocean and not the deep ocean. We've used the example of the last glacial maximum as this period of low atmospheric CO2 that seems to be related to this more intense biological pump, where these regions of excess nutrient concentration, which today are limited by iron, are more productive in the past due to greater dust supply of iron. Throughout Earth history, the biological pump is this key control on atmospheric CO2 because the ocean carbon pool is so large. Without it, atmospheric CO2 would be much higher than it is today, and we'd be experiencing a much different climate without it.